Hey everybody, welcome to Socratic Med's 15-week MCAT supplement course. We're on week 13. This is the second to last week. And today we're just going to talk a little bit about bioenergetics. This is going to be a short and sweet lecture. There's not too much we have to go over, but we'll just go over some, things conceptually that you definitely should know for the MCAT. So a little bit about the company. Socratic Med is a grassroots nonprofit that provides sensible solutions to students with disparate medical school opportunities. So I listed some of our contact information down here. If you're interested in learning more about the organization, you can look us up on Instagram. We have a YouTube page, our website. We use GroupMe to keep in contact with students that we're helping out. And at the very bottom, there's a link tree. So it'll probably have, the link tree will have links to YouTube, our website, and there are also signups for virtual office hours that we do, one-on-one virtual office hours with myself or any of the other tutors that we have here. And just on a personal note, my name's Chris. I graduated from Stony Brook in May of 2019. And I finished on a pre-med track with a BS in applied math and statistics. So I took the MCAT in April of 2021 and I scored a 520. For that test, it was the 97th percentile, and I got a perfect score on the psych-soc section. So my tutoring specialties are psychology, sociology, and also biology. And we do have multiple tutors, and each tutor specializes in something different. So we have, a bunch of staff at hand to help you with any subject that you need help with. So like I said, today we're going to talk about bioenergetics, really just a brief overview, because just keep in mind that these videos are supposed to be supplementing the 15-week course that we had previously done. So if you need clarification, you can definitely email me. I'll put my email at the end of the slide, but you can also go back to our page and just try to look through the original 15-week program, because there are a lot more videos on these topics there, and they could go a little bit more in depth. So these videos are really just aiming on filling in the gaps between the AAMC content and what we had already taught. So with that being said, let's just dive right in. So introduction to bioenergetics and ATP. So bioenergetics refers to the flow of energy through living systems. So the most commonly discussed on the MCAT are the anabolic and catabolic pathways of metabolism. So basically, everything that we've learned before: glycolysis, gluconeogenesis, beta oxidation, fatty acid synthesis, etc. Bioenergetics can also refer to growth and development, because obviously energy is important for growth and development as well. And one of the most important molecules in bioenergetics is ATP. So this molecule down here is ATP. We're going to go over it, I think, in 2 slides, but you could see right over here, we have this base over here, this 5-carbon sugar, and then we see 3 phosphate groups that are attached to each other. So we'll talk in a little bit about why ATP is important and how its structure allows it to carry energy throughout the cells. So energy sources, obviously all living organisms get energy from different sources. So plants, we should start with, it would make the most sense. They're considered autotrophs, but specifically they're considered photoautotrophs, which means they use sunlight to produce their own energy. Because remember, photo meaning light, auto meaning oneself. So just keep that in mind, that they use sunlight to produce their own energy. And this is how most of the energy actually comes into the ecosystem. Plants are, they use energy from the sunlight, just like this diagram to the right. And then it's consumed by one animal, that's consumed by another animal, that's consumed by another animal. So that's how these other animals, which are known as heterotrophs, which humans are, that's how they get their energy. So humans are specifically chemoheterotrophs, which means we utilize chemical energy by consuming other living things. So basically we just eat living things. We need to get our energy source from an organism because we cannot produce our own energy. Plants obviously, undergo photosynthesis to create their own energy, but we can't do that because we are chemoheterotrophs, so we have to get our energy from elsewhere. But plants and animals, once they do have their energy source, cellular respiration is the same thing. Plants and animals oxidize these organic molecules, these nutrients that they take in or that they create, and they give off CO2 and ATP. So ATP also stands for, is also known as adenosine triphosphate, and it's a high energy molecule that's often referred to as the energy currency of the cell. So most of the main goal of cellular respiration is to replenish ATP in the cell. There are a couple of other molecules, high energy electron carriers. There are other molecules involved in cellular respiration that are high energy and that can transfer energy, but ATP is pretty much the end goal because it's the most easily accessible. and it's the preferred energy for most cells. So ATP is used in transport of materials. It can be used in mechanical work, and it can be coupled with unfavorable reactions to drive them forward. So there are three major structural components of ATP, which is this nitrogenous base, if we look over to the left. So you can actually substitute, or your body can actually substitute this nitrogenous base to make different triphosphates. So for example, another common one, not as common as ATP, but another common one is GTP, which is used with G protein coupled receptors, for example. So different nitrogenous bases attached to these triphosphates will have different functions inside the body. So most of the energy in ATP is carried actually on the triphosphate chain. They're connected with bonds called phosphoanhydride bonds, and they are not the most stable. They're very high energy bonds. So hydrolysis of the outer phosphate usually is the most common mechanism of energy transfer. So when we hydrolyze, when we add water to that, we break that phosphoanhydride bond, and that third phosphate group pops off. That's where the energy release is coming from. So normally that phosphate will attach to something else, some sort of protein or an enzyme, and it's going to take the energy with it. And that's how we get this coupling of reactions. So if a reaction was unfavorable at one point, we take ATP, it's such a strong driving force that it actually has enough energy to drive an unfavorable reaction. So that's why ATP is super important. And like I said before, it's used in the transport of materials, so like in the cell, and it's also used for mechanical work. So ATP is important with those actin-myosin complexes with striated muscles, for example. So when ATP is hydrolyzed, it turns into ADP. So ATP is adenosine triphosphate and ADP is adenosine diphosphate. We're just taking one phosphate group out, and ATP is more stable and it's much lower energy than ATP. So it's not really usable. We can't really get a lot of energy out of it. The outer phosphate group is transferred to another molecule, but the cell does use the ratio of ADP to ATP to drive these sort of cellular reactions. So if ATP is too high and ATP is too low in the cell, it will start to produce ATP. It'll increase cellular respiration, will increase oxidative phosphorylation, and we'll try to generate some ATP with that ATP synthase. So the release of energy during the reaction, so ATP being hydrolyzed into ADP, meaning it can be used to drive forward unfavorable reactions that are vital to survival. So there are a lot of reactions that occur in your body that definitely would not happen without the help of ATP or without the help of enzymes. So if you look at this diagram at the bottom, you can see the coupled reaction. So we're adding H2O to ATP. That's going to hydrolyze it, and it's going to create ADP plus this inorganic phosphate. So if we look directly over at this first row, we'll see that the delta G, which again is the energy, but we'll talk about that soon in a little bit, is negative 7.3 kilocalories per mole. The reaction directly underneath it, we have glucose turning into glucose 6-phosphate plus water. So that delta G is positive, which means it's non-spontaneous. So that's not going to happen. Glucose is never going to just randomly pick up an inorganic phosphate because it's not a favorable reaction. However, if we have ATP near the glucose, we hydrolyze ATP, release that inorganic phosphate. ATP is so much higher energy and it gives off so much energy in that reaction that the net reaction, when we look right at the bottom overall, we're attaching that glucose 6-phosphate. We're attaching that phosphate to glucose to create glucose 6-phosphate and ADP. So these reactants, are a lot less stable than these products. So because we're combining these two reactions, we can actually add up their energies and we see that it is a spontaneous reaction. Again, because ADP and G6P combined have lower energy than ATP and glucose. So just because glucose 6-phosphate has a higher energy than glucose, but ADP has a much lower energy than ATP. When we add them together and we take the net, the overall reaction is favorable and it is spontaneous. So that is one of the super important jobs of ATP in the cell. Okay, entropy, enthalpy, and Gibbs free energy. So these are all some pretty important concepts when looking at the thermodynamics of the reaction. So we have a reaction coordinate diagram down here, which we'll talk about as well. We have starting reactants over here. This little hump Well, both of these humps are considered the activation energy of the reaction without an enzyme and with an enzyme. And then the products are over here. So we can see that the reactants are higher energy than the products. So this would have been, this reaction is releasing energy in the process. So enthalpy is defined by the following equation: H equals U plus P times V, where H is enthalpy, U is heat, P is pressure, and V is volume. So enthalpy is usually expressed as a change in enthalpy from start to finish of a reaction. So it's usually delta H equals delta U plus PV. But I don't really think you'll be seeing that specific enthalpy equation on the MCAT very much. They're not really, at least in my experience, they're not super concerned with you calculating enthalpy. It's more so using a given enthalpy to calculate the Gibbs free energy. So when pressure is constant, again, as it will usually be on the MCAT, the change in heat is equal to the change in enthalpy. So if we're looking at a reaction and the pressure is constant, if we come right back up here to our equation, H equals U plus PV, if we substitute that P, it would be a delta P because we're talking about the change during a reaction. If that delta P is 0, because the pressure remained constant, then this entire, this whole PV, this part right over here becomes 0, and H just equals U. So in that case, for all intents and purposes, enthalpy is equal to heat. And many times when you're given these values, pressure is held constant purposely so that heat is equal to enthalpy. It's a little bit easier for calculations, and just remember that when you're taking the MCAT, you're not going to have a calculator for it. So entropy is the disorder of a system. If you look up entropy, you'll find a bunch of really complex definitions and stuff that's really outside the scope of the MCAT. I mean, you're more than welcome to if that interests you and you want more background information about it. But basically, entropy is just the disorder of a system. So for the MCAT, your conceptual knowledge of entropy is a lot more important than memorizing it. So if we look right over here, if we look at this top row right over here, we're looking at ice cubes in a glass is low entropy. When we increase the entropy, we see that it changes a little bit. So we see that the state of matter, for example, will affect entropy. So going from a solid to a liquid, we're decreasing the order and we're increasing disorder. So whenever we increase disorder, we are increasing entropy. And if you can look on a molecular level as well, low entropy would be very highly ordered sort of structure. So say carbon structures, for example, they have very rigid formations. So those would be low entropy because they're very highly ordered. And then if we look at something, say, like a gas, where the molecules are just sort of bouncing around freely, that would be a very high entropy system because there's so much disorder. Okay, so those two, we're going to talk about Gibbs free energy next. So we can look at entropy and enthalpy individually to understand the system, but we're not going to get the full picture of it. Because when we see different reactions proceeding, we like to look to see if they're favorable. We say if they're spontaneous, then they're going to be favorable, which means if we leave it just out in the environment, leave the reaction to its own devices, it's going to proceed to equilibrium. And the way that we can calculate that quantitatively is by looking at this value called Gibbs free energy. So it allows us to measure the total free energy change of the system by incorporating the change in entropy and enthalpy. And the equation that we use, which you should definitely memorize for the MCAT, is delta G equals delta H minus T times delta S, where delta G is obviously Gibbs free energy, delta H is going to be enthalpy, and then that T times delta S is the ambient temperature times the change in entropy. So entropy, change in entropy is usually given if they're asking you a quantitative question that involves you calculating actually Gibbs free energy, they're going to give you the entropy and they'll probably give you the enthalpy as well. But most of the questions regarding Gibbs free energy on the MCAT are conceptual. So basically they're going to ask, they're going to show you something. They'll ask you about a specific reaction, and then they're going to ask you if it is spontaneous or not. And the way you have to do that is look at this equation and just sort of plug in positive and negative values and see what comes out for Gibbs. So with Gibbs, we can also identify the spontaneity of the equation, but it might not always be obvious. So when delta G is positive, when it's greater than 0, the reaction is not spontaneous. We call that an endergonic reaction. So it's bringing in energy. And it's not spontaneous. When delta G is negative, the reaction is spontaneous. And we refer to that as an exergonic reaction. Similar terminology to exothermic and endothermic, but don't get them confused. We can have an exergonic spontaneous reaction that is endothermic. And we can sort of more conceptually understand that by looking at this table right below. So an increase in entropy and a decrease in enthalpy are the two driving forces of spontaneity. So we'll look at the two simplest ones first. So if we look at this top row, we have delta H, delta S, delta G, and then comments. So if we look at this top row and delta H is negative, that means it's an exothermic reaction. It's giving off heat. So that is a favorable condition. When we have something giving off heat, that's what the reaction is tending towards. It wants to give off heat. It wants to get to a lower energy state. We have entropy as positive. That is also favorable. That's just how the universe works. Substances tend spontaneously toward greater states of entropy. So if we have something giving off heat, if it's exothermic and it's also increasing in entropy, we can even take those values to verify and plug them into this formula over here. So if we have a negative delta H and we have a positive delta S, that delta H term is going to be negative. This term, delta S, is going to be positive. So if we're taking a negative and subtracting a positive, this delta G is always going to be negative, no matter what that value of T is. So that's why whenever we have negative delta H and positive S, this comment over here, it's always going to be spontaneous. So we could look at that, and then we'll go down to the bottom row over here, where we have a positive delta H and a negative delta S. So the exact inverse. And it's the exact opposite. So it's not really favorable that a reaction is endothermic. And it's definitely not expected that the entropy would decrease. So if we have those two things happening, it's never going to be spontaneous. And again, you could plug it into the formula over there. If we have a positive delta H and a negative delta S, we're subtracting a negative. This term is going to be negative, so we're subtracting a negative, we're adding. So we have two positive numbers, and that's going to be positive. Delta G is always going to be positive for that. It's the other two conditions where you have to pay a little bit closer attention because they will ask you, the question that they may ask you is to identify the driving force of spontaneity. So when we have, for example, delta H positive and delta S positive, delta G can be either or depending on the temperature. So this delta H term, if we come back up to the formula, we're going to see delta G equals delta H. This is some positive value. We can say delta H is some positive value. And then this delta S is going to be positive as well. So we have a positive minus a positive. Now, the only thing that we are looking at after plugging those in is what is this T value? So if this T value is very low, it's going to make this whole term low, and it's going to be very likely that delta G is positive. If the T is really high, it's going to make this term much higher than delta H. We're going to subtract a huge number from a small number, and delta G is going to be negative. So that's at high temperatures. So that's why when we look back down at this chart, we see it's spontaneous at high temperatures, because once that T goes up, we're finally able to make that delta G negative. When they're both negative, we have a negative delta H term and then this negative T delta S term as well. And it's basically the same thing. So we're going to have negative and we're adding this positive term because we're subtracting a negative. So it's spontaneous at low temperatures because the higher this T gets, the more likely this delta G is going to be positive because we're adding this positive term back to this negative delta H term. So the higher this term becomes, the more likely it is that delta G is going to be positive. So that's why it's spontaneous at lower temperatures. And in that case, if we have a negative delta H and the temperature is low enough, we can see that this delta G is going to be negative and that this is going to have a spontaneous reaction. So since delta H is negative and we're giving off heat, it is said that enthalpy is the driving force of spontaneity in that case, because entropy is sort of playing against it. If we want a spontaneous reaction, we would really prefer that the entropy is increasing, but since it's decreasing in this case, we need a driving factor. We need this driving force to be so powerful that delta G is negative and that it becomes spontaneous. So in this case, if our enthalpy change is great enough, if we give off enough enthalpy, then that is enough of a driving force to create a spontaneous reaction. Okay, so the first question, A reaction is shown to have a negative value for delta H and a negative value for delta S. Which of the following statements is true? So take about a minute, pause the video. I will go over it in about 5 seconds. Okay, a reaction is shown to have a negative value for delta H and a negative value for delta S. Which of the following statements is true? And I put the formula right there, delta G equals delta H minus T delta S. So the reaction is spontaneous at low temperatures and the change in enthalpy is the driving force. So that's basically what I was just talking about. It's spontaneous at low temperatures because if we have this delta H term negative and this delta S term negative, we are going to have a negative term over here adding to this term, this positive term. So we have to keep this T low enough such that delta G does not become positive. So at lower temperatures, it's spontaneous. And because the entropy is increasing, really what's driving this reaction is enthalpy, is the release, that exothermic release. The reaction B, the reaction is spontaneous at high temperatures. It's not, because if that T gets too high, then delta G is going to be positive. C, the reaction is spontaneous at high temperatures and the change in enthalpy is the driving force. Well, the change in enthalpy is the driving force. But again, well, that should say entropy actually. So that would be incorrect both ways. It's not spontaneous at high temperatures and entropy is not the driving force. And D, the reaction is spontaneous at high temperatures and the change in entropy is the driving force. Again, all of those are incorrect. The only correct one is A. Okay, so kinetics and reduction oxidation, we are almost finished. We're just going to really go quickly over oxidation and reduction, probably something that we've all been learning since high school, but as I'm sure you've all noticed, when you get to organic chemistry and biochemistry, it becomes a little bit more complicated and sometimes a little bit more difficult to identify. So we should also talk really quickly about equilibrium. Delta G, that little mark above these deltas just means under standard conditions. So when we take standard conditions, we can calculate standard conditions, and it's very helpful for us to actually calculate the delta G in practice that's actually happening in the reaction. So delta G equals delta H minus T delta S obviously represents the equation for Gibbs free energy at standard conditions. So if we add a little prime there, which is usually what happens when we're talking about biology or medicine, so that prime is the free energy at physiological pH, because with standard conditions, all solutes are usually at 1 molar. But if we're talking about pH, hydrogen ions are not at 1 molar in the human body, definitely not. So we adjust to physiological pH and we just use that prime to mark, just to separate it from the regular standard condition delta G. But it's basically the same thing. You can use them you can't use them interchangeably, but these formulas, these next 3 formulas that we're talking about, anywhere you see that delta G standard, you can use that physiological pH delta G as well, and the formula will work. So we can relate the free energy of a system to its equilibrium constant. So that delta G, if you can't get it from this formula, delta H minus T delta S, you probably won't be able to, because a lot of times they won't give you the actual standard number for enthalpy and entropy. You would use this equation, which is negative RT ln KEQ. So you should probably memorize all of the formulas on this page. It would definitely be helpful. You will probably have to use that formula. So our negative RT ln of K equilibrium. So R is the universal gas constant, which will probably be given to you on test day. There are a couple of them depending on what unit you use. And then you just multiply that by the ambient temperature in Kelvin, obviously. And then you take the natural log of the equilibrium constant, which we denote as KEQ. So KEQ is just the concentrations, the product of the concentration of the products over the product of the concentration of the reactants. So KEQ, this second one, to be a little bit more correct, there should be subscripts on each of these concentrations. Outside of the bracket, there should be a little EQ, just like there's on the K. But it's the same formula. You would plug in the concentrations of the products and then the concentrations of the reactants to get the equilibrium. So it's basically just the ratio of products to reactants once the system reaches equilibrium. So when we know that, we have all this information, we can actually calculate delta G of a reaction that we're running. So delta G is going to equal that standard delta G plus RT ln of Q, where Q equals C times D over A times B. So it's the same thing, it's product over reactants. The only difference between Q and K equilibrium is that K equilibrium is that ratio when the system is in equilibrium, and Q is the ratio when the system is at any other point. So Q can equal KEQ, but for most of the time, it's not going to. So if we take the natural log of that, multiply it by the gas constant times the ambient temperature, and we add that to the standard delta G, we are going to find the actual delta G for the specific reaction that that we're looking at. And it's just important to keep in mind that when calculating equilibrium constants, you do not use solids or liquids because they have fixed concentrations. So they're not going to be changing throughout the reaction. Water is not going to be changing. If you have, some sort of solid that's in the water that precipitates out, that's not soluble, it's not going to change concentration. Aqueous solutions will change concentration, and so will gases. So those all have to go in the equilibrium equation. Okay, so we were talking a little bit about thermodynamics, and kinetics obviously are also super important for finding out information about these reactions. So thermodynamics will tell us about overall energy changes and the relative stability of the product, but not about the actual rate of the reaction. So when we have spontaneous, we can look at that delta G equation and we can decide if the reaction is spontaneous. So if it's spontaneous, delta G is negative, it's going to tend towards its equilibrium, but it will not, those formulas do not tell us anything about how fast it reaches equilibrium. So we could have a spontaneous reaction that is occurring over hundreds or even thousands of years, or we could have a spontaneous reaction that can be completed 500 times in 1 second. We have to look at kinetics and reaction rates in order to actually get this information. So for a reaction to move forward, the reactants must receive an activation energy to reach the transition state. The transition state is a high energy unstable form that quickly disappears as the product forms. So if we're looking at this diagram, this is called a reaction coordinate diagram. On the y-axis, we have our energy level, and it's very intuitive. So the higher point you are, the higher energy, the lower point, the lower energy. And then it's the reaction coordinate, which is basically, we could basically just say it's sort of the time of the reaction, or how the reaction proceeds forward, to be more accurate. So we start with the reactants. If we can get activation energy in there through whatever method, we have enough to bump this up. We'll change the molecule just slightly and super, super quickly. It's very transient. It's going to hit its transition state. It's very unstable. And because it's unstable is why this reaction proceeds forward. So it's going to tend toward the products because the products are much more thermodynamically stable than the reactants. So if we can get it right up to this hill, it's going to roll down that way because it's a much steeper fall. All products, not all products, but all reactions, basically the goal in terms of energetics is to get low energy products. We're always moving from high Gibbs energy to low energy. That's just how the universe works. So the transition state is unstable. It quickly disappears as the product forms, and catalysts work to lower the activation energy rate. So the rate of reaction increases. So basically what affects how quickly the reaction will proceed is how much of this activation energy that we actually need. So if it's a higher hump, we're going to need a lot more activation energy. And it's going to take a longer time for these reactions to proceed forward. If we can somehow lower the activation energy, then it's going to be a lot easier to impart energy into the system, and it's going to be a lot easier to drive the reaction forward. And that is what catalysts do. So we have enzymes in our body. Those are the catalysts that usually work at our physiological pH. And what they do is they just bring this activation energy down. So these reactions can proceed much, much more quickly. So catalysts do not affect the thermodynamics of the reaction. Keep that in mind. So if we add a catalyst to this system, we're going to take this bump, we're going to take this hump a little bit down, a little bit farther down, but where the reactants and the products are on this reaction coordinate diagram are not going to change. The actual change, the difference in energy, products minus, or reactants minus products, is always going to stay the same. It doesn't matter how many catalysts we add to the system. So oxidation and reduction, humans store energy in reduced molecules like fats and carbohydrates. So if you ever hear reduced, if a molecule is highly reduced, it's going to be carrying energy. It's carrying high energy. If something is being oxidized or something is oxidized, it is probably lower energy. So if you're oxidizing something, your body oxidizes things to take energy, to transfer energy from that substance to something more usable. And that's basically all cellular respiration is. So as the molecules proceed through cellular respiration, they're oxidized. The energy given off during oxidation powers the regeneration of ATP. So most basically, oxidation is the loss of electrons and reduction is the gain of electrons. So since energy flow follows electron flow, which is something definitely helpful to keep in mind for the MCAT, cellular respiration oxidizes the nutrients absorbed in digestion and transfers this energy to molecules like NAD plus and FAD, which will then use these electrons to power ATP synthase. So that NAD plus and FAD, we refer to them as high energy electron carriers because they're taking these electrons from the nutrients that we're absorbing, and they are carrying them over to the electron transport chain so that we can use this high energy to spin that ATP synthase wheel and couple ADP and inorganic phosphate. So oxidation may appear as a gain of oxygen atoms. It may appear as a loss of hydrogen atoms or the loss of electrons. So technically all three of these are, it is in the process of losing electrons. It's just not always as obvious as it may have been in general chemistry. So if you gain an oxygen atom, oxygen, you can sort of think of it that oxygen is very electronegative, and it's taking electrons away from the original molecule that it is being added to. So technically that would be considered a loss of electrons. So when we're adding oxygen atoms, or when we're adding bonds to an already existing oxygen atom, we are oxidizing it and we are lowering its energy state. Same thing with the loss of hydrogen atoms. If we're looking at NAD plus, that cofactor, if we have NADH and we get rid of the H, we have NAD plus. So it's losing a hydrogen atom, but it's also losing that electron in the process. So that would be considered oxidation as well. It's going from a higher energy state to a lower energy state, or the straight loss of electrons. So if you're looking at a metal, for example, and it's changing an oxidation state and it just loses an electron. So iron, for example, if it goes from Fe2 plus to Fe3 plus, it's losing an electron, it's being oxidized. Reduction may appear as the loss of oxygen atoms, gain of hydrogen atoms, or the gain of electrons. So the exact same thing. Loss of oxygen atom or the reduction, the loss of oxygen atoms or the lessening of the amount of bonds on a specific oxygen atom. So if we go from, say, a carboxyl group to a hydroxyl group, we're removing a lot of bonds from oxygen, and technically we're reducing it because we're adding electrons to the molecule. Gain of hydrogen atoms, we can look at NAD plus again. If we add an H, it's a hydride ion. It's not an H plus ion. So that means it's actually carrying an electron. And that's how we get to NADH, which is that high energy reduced form. And the gain of electrons, same thing. If we're looking at a metal, for example, or if we're looking at any sort of ion, anytime it gains an electron and changes its oxidation state, that would be considered reduction as well. Okay, so this is our last review question. Which of the following constitutes an oxidation reaction? Take a minute and a half, 2 minutes, read these, and we will go over it. Okay, so which of the following constitutes an oxidation reaction? So the formation of a disulfide bridge, the formation of ethylene from ethane, NADH converting to NAD+, and changing the hydroxyl group on a molecule to carbonyl group. So the correct answer is D, 1, 2, 3, and 4. These are all oxidation reactions. We're going from a higher energy state to a lower energy state, and we are losing electrons. So let's take a look. The formation of a disulfide bridge, that's a good one because when we're talking about biochemistry, this is a molecule and this is a sort of interaction that always comes up in the context of redox reactions. So if you have two sulfhydryl groups, which is an SH at the end of it, they have that hydrogen on it, they have to lose those hydrogens in order to form a disulfide bridge. So those hydrogens are going to be lost and they're going to take an electron with them because we have to have an empty orbital in order for those disulfide bridges to form. So they have to share those electrons. So they're losing electrons when they form those disulfide bridges. So that would be considered oxidation. The formation of ethylene from ethane, all we're doing is yanking a hydrogen out and we're forming that double bond. Same thing, we need to take electrons out in order for that pi bond to form. So that would also be considered oxidation because it's losing an electron. NADH converting to NAD+. we are losing that hydride. It's taking an electron with it, and it becomes plus because it lost an electron. So that's oxidation as well. And then we have changing the hydroxyl group on a molecule to a carbonyl group. So hydroxyl is a single bonded OH to a carbon, and then the carbonyl group is a double bonded oxygen on that carbon. So if we're adding a bond, we're getting rid of that H and we're adding a bond, that would be considered oxidizing, because remember, we're adding more bonds to the oxygen molecule and we're losing electrons. So 1, 2, 3, and 4, those are all correct. So kudos to you if you got that one. And that's pretty much all I have for today. If you have any questions, please email me right there, Christopher Scagnelli1. If you have any questions about the lecture, about Socratic Med in general, if you want any MCAT tips, if you want to be added to the GroupMe, don't hesitate. So next week is going to be the last week of the 15-week MCAT supplement course. So I hope to see you guys then. Happy studying until then.